Thank you very much. And I'm just wondering whether to sit down or to stand up. Now, normally you have to stand up for lectures, don't you? But when I joined a geography department, I found that human geographers don't. They just sit down. And it's very relaxing. But does it provide enough of a zap and pizzazz? I don't know. The big advantage is that I'm not going to get in anybody's way if I'm sitting down, which is a point. I'll start with that. But if you disapprove, just start shaking your head gently, and then I'll run around a bit and see how we get on. So I was so delighted to find that rock face on the left. It's a rock face about this big, and it looks just like this. It looks for all the world like a fossilized hand, but it's a Precambrian rock. So that isn't the course of future evolution foretold. Stromatolites are not soothsayers, but that's just to illustrate one of the remarkable examples of their morphology. That sort of morphology actually is called digitate. So it does refer to the finger-like shape. They quite often find these growths in that way. So stromatolites. So literally, stromatolite means layered rock. And Kalkowski in 1908 thought of this term, but of course it isn't used for layered rocks in general. It's for rocks which have some particular attributes, and we'll have a look at them. And early in my career, I was interested in them. I even did an undergraduate dissertation on them. But through my career, I've just been at the periphery of stromatolite research. I haven't focused on it intensively, and I'm pleased I haven't, because it's proved so difficult to understand the morphology of these things. I'll show you lots of things, and there's lots of interesting things, but the central question about how do you explain the shapes that they make is one that is mostly too difficult to answer, although there have been some useful developments, which I'll tell you about. So stromatolites then are layered rocks. They're created by an interaction between microbes and the environment. And you find them today, and you find them in the deep time, particularly in the Precambrian, before other animal groups evolved. They're particularly abundant there. And we also find lots of microfossils in the Precambrian too. But the stromatolites, they're macroscopic. You can go to an outcrop and you can see them. And so they are, for most of the Precambrian, the only macroscopic evidence of life that we see. And you see that right back to the Archean, back to 3.5 billion years ago. However, they're not simply fossils. So what are they? Now, I wasn't expecting to have an internet connection here, but if you look up on YouTube stromatolites, this is a brilliant 2-minute introduction to them, which is really nice. I can't play you that now, but that's worth listening to. So my title is about making mountains out of microbes, and so I'm exaggerating a little bit, but certainly you can have very thick cliff faces full of stromatolites, and what we've got here is like a stromatolitic reef. You see these wiggly layers here, and they die out over here. This is smoothly laminated normal sediment, and this is a stromatolite. So it's a bit like a cross-section through a coral reef. So they form reef-like structures in places, and these can persist upwards for many, many tens of meters. So you can see the idea. You can make mountains out of these sort of things. And here in East Greenland, we're down the Precambrian, and quite a lot of the thickness of carbonate rocks we see there are made of stromatolites. And if we look at individual ones, we've got these lovely rounded ellipsoidal shapes here. These are bioherms, just like the small reefs that you see in the Phanerozoic record. And they're rather nice. And then you zoom in and look at them close up, and they've got lots and lots of small domal structures. So they on a small scale, lots and lots of little domes. Large scale, these kind of ellipsoidal shapes. So there's no simple answer as to why they should form an ellipsoid, but they do. there's probably some basic bit of physics, and there have been some papers in physics journals actually trying to explain some very simple parameters why they get the shapes they have, but I'm not aware of any papers that have really shown why the ellipsoid is a particularly common type. So a biome like that would have grown probably in shallow subtidal conditions, whereas intertidal ones will be typically flatter. And there's a close-up of some of these things. Now, some of them have got really very clearly developed layering, and it's picked out by dark layers and light layers. And that's because these are limestones that have been partly dolomitized. The dolomite's got a bit of iron in it, and it gives it this pale weathering, and whereas the limestone is the dark weathering. So that's quite common. In the Precambrian, most carbonate rocks are dolomitized, because there's a lot more magnesium in seawater in the Precambrian. So dolomite is very, very common. And here they're partly dolomitized, and it's showing up the structure really very nicely. So they're mysterious, aren't they? They're forming these growth structures with distinctive morphologies on different scales. And here we've got the equivalent of those Greenland rocks in Svalbard, and that is the similar age rock to the biomes I was showing you just now. And this stuff, this white stuff, has got the most beautifully preserved microfossils that I'll show you later. It's a formation called the Draken Formation, and I'll show you some examples of those microfossils later. So here's Richard as a fourth-year undergraduate coming on the expedition. We did tell him no personal specimens on an expedition, Richard, it's not allowed. And I think eventually I managed to prize this specimen off him for the collections of the Lapworth Museum. But it is beautiful, isn't it? Again, we've got this differential relief there from dolomite and calcite layers, and you can see the individual growth structures. We call these columns when they're separate, or if they're all joined together, we call them domes. So we've got columnar structure in here. And when you start looking at it, you start wondering about just how distinctive are those shapes. I mean, if those were corals, for example, you'd be looking to get a very distinctive shape, and then you'd be wanting to give it a name and to compare it with other examples. It's very, very tempting to want to do that. And that's actually what people have done in the past, as I'll explain in a moment. Now, they form by an interaction between microbes and the environment. So they're not fossils, and they're not sedimentary structures, because you need the organic activity. They're what we call organosedimentary structures. So they're in the same box, if you like, as trace fossils. Now, trace fossils, of course, are the records of activities of animals, and so they form very distinctive burrows. So you can actually give these, these are given names. They're given names for the shape of the burrow rather than the organism, but they have distinctive shapes, and there's a direct link to the organism, but they represent a record of behavior. So that's an example of a type of organic sedimentary structure, trace fossil. Stromatolites are similar in that sense, that they're not fossils and not sedimentary structures. But how do they form? Now, in the 1950s, the Russians got onto this, and this is Misha Semikhatov, who became a global expert on this, and he and his colleagues reconstructed the shapes of these structures in 3 dimensions through painstakingly taking serial sections and building up these pictures, and then they described them as genera and species. So they actually built up a taxonomy, just as you would for any organism. But was that valid? One of the issues here is that you can reconstruct something like that, but actually at any one time, only part of the structure would be above the level of the surrounding sediment surface. That's what we call the synoptic relief. So the synoptic relief is much less than the total height of the structure itself. And you can sometimes determine what that synoptic relief is if you can find layers that you can trace from one of these columns into the ordinary sediment in between the columns. So geometrically, they're very complex. This is a summary of many different descriptions, and there are lots of different distinctive shapes. They've all got names, but are those names valid? Now, back up in the 1980s, 1990s, even up to the early 2000s, there have been some Australian and some French workers who continued this tradition. And one or two of those names are still used, but mostly people have said, we can't do it. It's too difficult. They're not fixed in morphology. They're not fossils. So we've got, so all that work that was done, there's a lot of insight there, people getting their eye in for these structures. But if they're not simply organisms, then that must represent something else. It must represent some very distinctive interaction between the organism and the environment. But what? It's very difficult to tease that out. So people name lots of these different things, and this is the diversity of different genera, as they were called, or species. And they seem to be most diverse near the end of the Precambrian, near the end of the Proterozoic, around a billion years ago, and then becoming less diverse as you go into the Cambrian. They're much less abundant. So, and people generally haven't attempted to name the ones that occur since the Cambrian. So there's a great body of work, and it's kind of now, it's like a backwater in history now, because they got so far, they became expert, but they weren't able to convince everybody else that actually what they were doing was the right thing. So it's very interesting. I think there are quite several lessons about research in general that one can glean from what stromatolite workers have done, as we'll see. Now, stromatolites can be fun, as you can see here. Here they are, they might be in Vogue magazine or something. They're showing off their arched layers. And during the 1970s, when I started research, there was a tremendous amount of interest in stromatolites. And this book was published, very thick book, the first time there'd been a substantial publication about them in 1976. And Elsevier published it. And they charged the outrageous price of £64. And everyone at that time, it was just so astronomical. People could hardly believe that they dare charge that much. But university libraries bought it, if nobody else. So that was a lot, and that was the first time that actually Western workers and Russians published in the same volume, and there was actually communication going on, which is a really good thing. So I got involved with this, and we thought it'd be good to try and have an international project to try and continue this cooperation and have field visits and so on. So I did a talk about it, and then as a result of that talk, someone came forward to lead the project, and this is Bob Byrne here, and the project emerged as an international geological correlation program project, which is basically a really good initiative about bringing different countries together and getting people to talk to each other. And for quite a while, there was this stromatolite newsletter published, which kept everybody up to date with that. But nevertheless, as the eminent sedimentologist Kerry Kelt said, this is a beautiful comment, I think. Stromatolites are a traditional mystery. That's kind of a really profound statement. That is, they are profoundly difficult to understand, and he expressed that in that way back at that time when everybody else was getting excited and thinking they could solve all the problems, and actually it's really difficult. Well, let's go back to the beginning then, and back in 1825 James Hall first described some laminated structures that we now know as stromatolites, and he found them in Saratoga Springs in New York, and they basically look like overgrown ooids. It looked like an oolitic or pisolitic limestone. But when somebody went back there a few decades later, they realized that actually they weren't spherical structures. They were just looking at cross sections through some of the domes. So actually in 3D, there were things like this. So in cross section, they look like that. And so they started to give them names, and this one was called cryptozoan, which kind of means hidden animal life, which of course they're not animal life, but anyway, that was what was thought at the time. In the 1960s, people started saying, well, we think we got some modern ones of these peculiar things, and in lakes, there are these structures which are hard encrusted structures that grow up, and they're covered with a microbial slime. And these structures were thought to be, oh, these look quite similar to these things we're seeing in Precambrian rocks. The difference is that this is freshwater, whereas the Precambrian ones are all marine. So something had changed in between. So in the 1960s, then people started sort of giving them names. So we had Collenia for things that were what we now call domal stromatolites, and the cryptozoan was the ones that we'd now call columnar stromatolites. And they are really very beautiful. I was very lucky to be able to visit the stromatolites in Glacier National Park in Montana, where they're beautifully developed in the Mesoproterozoic. That's about 1.5 billion years of age. And there are all sorts of really intriguing things going on here. You know, they're growing up at an angle here, and then they're starting to accrete in that direction. Now, why is that? And there are two types of hypotheses for how it is that they can change direction as they grow up. One is that they may that it may be something to do with light, and you may get faster growth in the direction of light, and sometimes there can be some disturbance of the bed which would cause that growth to change. Or it could be something to do with the supply of sediment. If sediment is coming from a particular direction, that may help them to accrete in a particular direction, perhaps. So all sorts of mysteries. And you've then got the issue about, you know, how distinctive is this shape, and is this diagnostic of anything? So working on modern environments would be a good idea. But in fact, it was had been done decades before this was started. This is a guy called Maurice Black, who spent most of his career in the University of Cambridge. And late in his career, he became very famous because he first photographed coccoliths in the chalk. The coccoliths are the chalk is made of coccoliths, these tiny fossils. Plant microfossils, and he had one of the very early scanning electron microscopes in Cambridge and first described them. So that was a great thing he did in the 1950s. But back in the 1930s, he was a very young man, and he went to the Bahamas and worked on Andros Island in the Bahamas and identified these microbial mat structures. I've just got an aside here. I've got another anecdote, and the anecdote is to do with the peculiarity at Cambridge. It's a very odd place. So Maurice Black worked on a PhD. He did all this stuff in the Bahamas. It was fantastic work. He got a lot of it published in thick papers. It was brilliant. It was worth a PhD. But he didn't get his PhD. And why? Because his supervisor, O.T. Jones, was away for a term. And Maurice Black says, I'm not paying my supervisory fees for that term when that guy wasn't here. And the university said, well, in that case, you're not going to get your PhD. And he said, so be it. So for the whole of his career, he was only Mr. Black. He wasn't Dr. Black. But actually, everybody thought he was so wise that they called him Dr. Black anyway. However, there was a genuine Dr. Black in the same department, Bill Black. But Bill Black didn't have his degree from Cambridge. So in the very stuffy official letters within Cambridge, he was always addressed as Mr. Black because he didn't have a Cambridge PhD. So all very confusing. Anyway, Maurice Black did this work in the Bahamas, and you can see that you get on the surface of the flats there, the tidal flats, these strange polygonal structures. And these are the kind of leathery mat made of secretions made by bacteria, what used to be called blue-green algae. and they actually trap sediment as well. And you have layers of sediment and layers rich in richer in this organic matter of this microbial mat. And he identified these different sort of morphologies that they form. So we're saying, well, look, this is something like this is telling us a bit about some of these Precambrian structures that we're seeing. Now, we're not seeing the great diversity that we see in some of the Precambrian ones I've already shown you. But in terms of processes, in terms of how these things can form, the idea of sediment trapping and binding by a sticky mat, and having layers richer in organic matter and layers richer in sediment, that idea came from this work. So we see these kind of microbial mats in all climates. They're in Antarctica, they're in the tropics, they're in the Lucker Estuary in South Wales. So here we are on in the supratidal flats there, and these things, when you pick them up, they're slightly cohesive, they're slimy, and they're this wonderful gelatinous material that the cyanobacteria secrete. So as I say, when I was a student, they were called blue-green algae, but biologists said they're not algae. They don't have a nucleus in the cell. They're prokaryotic. They must be bacteria. And because they're blue-green, we'll call them cyanobacteria. And that's the name that's now given. So instead of calling them an algal mat, as they used to be called, we now call them microbial mats. And these cyanobacteria form either coccoid cells, round cells in clumps, or they form a long line of cells to give you a filament. And you get both types, and you get a whole assemblage of different species within a mat. And they photosynthesize, and sometimes you can actually see this. You can see here, these are the oxygen bubbles forming on the mat from photosynthesis. So they are converting CO2 to oxygen.
What's that? Where did he
now, deeper in the mat, in fact, there are other bacteria come along, and they produce hydrogen sulfide, and they do all sorts of other things as well. But at the surface, you get this oxygen production. So this is an example where they've you've got a mat with a whole series of different layers, and some of them are reducing and produce H2S. There are layers that have got calcareous precipitates in them because of chemical reactions that are occurring within this stuff that is secreted by the algae. So the simple acronym for this is EPS. It stands for extracellular polymeric substance. EPS is how it's usually referred to. So it's the stuff that they secrete, and they secrete it in order to protect themselves. It stops them drying out. It stops them being damaged by ultraviolet radiation. So it's great stuff. And it's also a fertile medium for mineral precipitation. So calcium carbonate can often precipitate in them, and all sorts of biogeochemical things happen within there. Now, if we look at the overall geological history, well, the Earth is formed down here, 4.6 billion years. And then, of course, we're getting sedimentary rocks coming in the Archean. And around 3.4 billion years, we do see some apparent bacterial fossils in stromatolites in Western Australia. So the oldest stromatolites and the oldest potential fossils are around this time. And then we then get oxygen buildup much later. We have some climatic disturbances with these snowball, with first snowball Earth, and then much later other snowball Earths. And in the meantime, we start getting some eukaryotic organisms which do have a nucleus in the cell, and then some organisms that have multiple cells as well, including bacteria. So these things happen, but it all happens very, very slowly. But all through this time, all through these billions of years, it's only the stromatolites that are there as the macroscopic evidence of life. So for most of the geological record, that's what you find. That's the evidence of life of the stromatolites. And then we then get the Ediacaran fossils coming in right at the end of the Precambrian. So now, there was a great breakthrough when they first discovered the fossils inside these things. And the guy that first described them, well, it's Barkhorn and Tyler. Barkhorn was a professor in Harvard University, and they worked on something called the gunflint chert, which is a banded iron formation with chert layers and iron oxide layers on the banks of Lake Superior. And here it is in the field. And when they looked in thin sections, they could see these little micro stromatolitic structures. And inside them, when they enlarged it right up, that's 20 micron bar, 20 thousandths of a millimeter. And you can see the individual filaments we're looking at are only a micron or two of, thousandths of a millimeter across. So they're really, really tiny structures, but they found them beautifully preserved by early silicification soon after sedimentation in this structure. So suddenly, Precambrian paleontology was born. This is 1965, having not found any body fossils at that time. Then suddenly there are body fossils, and they're microfossils. And of course, there's now a huge array of microfossils have been found in Precambrian rocks. They're not commonly found, but and often you have to look in thin sections and so on. But they're there, and actually that helps to establish the biogenic origin of structures, of stromatolitic structures. So generally then, these stromatolites that we're looking at formed by some combination of trapping of sediment, which is shown there, or precipitation of calcium carbonate, which is shown here as these structures. And sometimes that's specifically in association with microbial, with this EPS, this extracellular polymeric substance. So, and each one you look at has got a different combination of factors responsible for its formation. And it's not always immediately obvious, and you sometimes have to use special techniques to work out what is actually forming the different layers. Now, up to now, then, so I've been leading you down the garden path because I've been telling you that they're called layered. The word stromatolite means layered rock. Everything I've shown you has got layers in it. And I'm now going to show you some things that don't have layers in, and it's still in the stromatolite nature. So I hope you don't feel that this is wrong, but there are things that form in a very, very similar way which don't have layers. And they tend to have a clotted structure. So as in a thrombosis, they're called thrombolites. So what's going on here? Why do we get things? So these are things that are formed by microbes, but they just don't have layers. And so it just represents a slightly different type of community of organisms. And here's some modern ones that I found in Australia. This is really, they're in a lake and they're precipitating monohydrocalcite, which is probably a mineral you've never heard of. And I don't think I've ever heard of it anywhere else, but it's calcium carbonate with a bit of water in it. And it doesn't last very long. It changes to calcite. But the microbes there, you can see that they're responsible for the green color. They form a slime on the surface. The calcium carbonate precipitates underneath it, and they grow up as these little knobbly structures. So this is an example of a modern thrombolite. And thrombolites can be found in various lakes. Here's some other ones in Australia. Oh, this is one that I visited Lake Clifton in Western Australia, and I think I can get that to it. Yes, here we are. So you can see the water moving there, and these are oh, that's a very short video clip. There we are. That was it. You had it. So you can see that when the water level drops, the structures stop growing. So they can't grow above the water level. They grow up to the water level, and then they have that flat top in that case. So thrombolytic structures are actually quite well developed today. Now, they tend to have a more complicated community of organisms in them than the stromatolites. They tend to be more other types of, they tend to be algae and some protozoans and other things in there. So they're not as simple as the microbial mats that form. Right, so it's gone off, that form stromatolites. Let's go to the next slide beyond that one, if that one's a problematic slide. Okay, so I can, oh, okay, it's on the screen on my computer, but it's not on your screen. Oh, there it is. Yeah, we're okay. Excellent. So there are lots of different processes that go on then, and one of them is trapping and binding of sediment, and that's at the top of this diagram. Another one would be inorganic precipitation of cement. Another one would be biologically influenced calcification. That's when these calcium carbonate grows in the EPS. And so the things I'm talking about belong on that triangle. But whereas skeletal calcification, that will be like a coral, that will be like a recognizable organism. So we're not in that bit of the diagram. We're in this triangle here. And if you look on that triangle, you can put different locations in different places on that. So that the lake I've just shown you, Lake Clifton, that would fit over here. It's more strongly biologically influenced calcium calcification rather than inorganic, and there's not so much sediment. The most famous location for stromatolites is Shark Bay. I'll show you that in a minute, and there's more sediment in that case. Now, so this diagram, that tetrahedral diagram, this triangle here is telling us about the different structures that can be created. They have different processes going on. And the guy earlier, I showed you a picture of Bob Byrne, who was running that project. And he and his student, Linda Moore, came up with this brilliant new term they called microbialites. They said, well, they're made of microbes. Let's call the rocks that they make microbialites. So that includes the stromatolites and the thrombolites. So I could have called my lecture microbialites, but you might have turned, I don't know what you just said to that. But anyway, nearly all of it is about stromatolites. So there we are. Now then, and the next thing I want to mention is, why did they decline? They declined very radically at the end of the Precambrian. What changed? Well, the most likely explanation is it's the advent of organisms that eat microbial mass. And one organism that's very good at it are these sericea gastropods here. And I think I took this picture on a modern tidal flat in England, or possibly Wales, and you can see they're just having a whale of a time feeding on the sediment, eating it and excreting the sediment. And so they stop the mats being as fully developed as normal. However, those sorts of organisms cannot grow very easily if the water is very saline. So in a hypersaline marine environment, you've got more chance of forming a stromatolite. And that's indeed what is found in this location of Shark Bay in Western Australia that I'll tell you about in a minute. However, before we get there, I just wanted to say, having introduced the topic of the grazing and so on, and the fact that it's much more difficult to be a stromatolite after the Cambrian, where have we some? In the British succession, and there are some classic ones. The Cotton Marble, the late Triassic lagoon, right at the end of the Triassic. This is this amazing, extraordinary stuff. So some of this is thrombolytic and some of this is stromatolitic. And then there are stromatolites that grow around tree stumps in the Purbeck in Dorset. So there's a couple of examples. And here's one from the Jurassic of Skye. That's a thin section, and you can see it's made of grains that have been bound together. So there are Phanerozoic examples, but they're just not nearly as well developed as the Precambrian ones. So in my PhD area, then I was working on this stuff on the Isle of Islay in Western Scotland, in the Dalradian, in the Precambrian, and there's a big stromatolitic body there. This was me at the time, a bit more hair, as you can see. And this was in a formation called the Bunhaven Formation, which lies above the famous glacial deposits of the Portasque Formation. And I followed Tony Spencer, and Tony's paper here, it's got these lovely drawings of these stromatolites showing you their shapes. You've got the bioherms like this, and the biostromes, which would be the continuous beds. So, for example, this bed here shows you fairly flat layers. That's this one, with just a gentle dome. And then as soon as you get sand coming in, which is here, everything gets broken up and you get these columnar structures forming. So there's a very clear link between the environment changing and the morphology changing. in that particular case. So the environment becomes much more energetic, and the map becomes less continuous and more partly eroded. So so that's nice. So there is that sort of link. I mean, it's not a very sort of fundamental understanding, but it's a general understanding that the environment is changing the characteristics of the stromatolites. And one of the things I did there was to reconstruct. There's lots and lots of outcrops dissected by faults, and if you undo all the faults, you end up with this is the geometry of the stromatolites. The stromatolites are shown in black, and you can see how they change laterally, and you can get some idea from that of the lateral persistence. So the average lateral persistence is a few kilometers based on this data. And I saw 3 adders in one day in May. That was rather special. I didn't used to like adders, but I've got used to them now. I think they're great. Now, one odd thing was that when I was working on these rocks, which are very shallow water, tidal flat type environments, subtidal, tidal flat, I didn't see any cycles in them. So, if you've done any reading much about geology, geologists often talk about rocks being cyclic and having patterns and cycles. And in the case of tidal flats, then the obvious thing are things like the Arabian Gulf, where the shoreline builds out and you go from subtidal to intertidal to supratidal deposits, and then everything subsides, and then you repeat. And a number of geological successions are like that. But I didn't find that, and I did statistical analysis and said, well, this doesn't work here. Something, that's not the pattern. It can't be simple linear fasces belts. And in fact, later, after I'd finished my work, Brian Pratt and Noel James came up with the tidal flatten island model, which had a different geometry, whereby instead of having fasces that were lines, which give you these cycles, you actually get more spotty texture with little islands here, there, and everywhere, so that when you build up a succession, you don't get cycles. So that was interesting to see that. And finally, now we got to Shark Bay. So Shark Bay is a hypersaline inlet into the coast of Western Australia. So here is a bank of seagrass that restricts circulation into the bay. And the salinities in the bay can be up to twice normal marine salinity. And so what that means is that invertebrates are not allowed in. So you can think of the seagrass as the bouncer and the nightclub, and you're not allowed in unless you are a microbe. And so, and this was the famous place that in 1961, Brian Logan, a young research student, published this paper about these intertidal stromatolites in Shark Bay, which you can see on the shorelines. They form these club-like bodies, and one of the things that he showed was that these things form primarily by trapping of sediment. So up to then, people had often said, well, we think some of these things form by precipitating calcium carbonate. He said, no, these are modern ones. They're intertidal. In fact, he says very strongly they're intertidal, and they form by trapping sediment. Now, it turns out that he was really very wrong in some important respects. But when I was a student, this was the dogma. So as I said, I was writing a dissertation about these things based on the literature. And my lecturer was Dick Aldridge in the 3rd year, wonderful man. And he said, stromatolites are intertidal. And I said, no, they're not. Because he was taking the textbook view, of course, because he wasn't an expert on them. But so that's what people thought, and it was based on what they saw in Shark Bay. And Brian Logan was responsible for really pushing that. And so you have what ends up as a meme. So this is some artist's idea of early Earth, and they're the stromatolites, and they look just like the ones in Shark Bay. However, that's very misleading. Now, the person that discovered the Shark Bay stromatolites first was a guy called Phil Playford. And he actually showed them to Brian Logan, but Brian Logan went ahead and worked on them and published them. Meanwhile, Phil Playford was publishing on this reef in the Devonian in Australia, and it's just stromatolitic reef. Now, there's no way that that can be intertidal. I mean, it's a great reef structure. It's tens of meters thick and so on, and it's between lagoonal sediments and deeper marine sediments. So clearly there's a bit more to it. And then what about these things? These are strongly aligned structures that are formed. This is an example in Canada, the Proterozoic in Canada. And they seem to reflect, large-scale tidal alignment in subtidal channels and so on. So that what's going on? And then people started saying, well, look, we've got these stromatolites that are forming in between islands, in some depth of water in the Bahamas. So clearly they're not restricted to intertidal environments at all. They're clearly growing. They're growing there. Yeah, so this is a 10 meters depth, for example, these things. Yes, but Brian Logan found them in the intertidal environment, didn't he? Well, actually, when you look in the subtidal environment, there they are. And actually, that's where they grow. So these big club-shaped stromatolites in the intertidal zone are relics. The sea level has relatively fallen in that local area. They grew when it was covered in water. They're now stranded in the intertidal zone. It wasn't where they grew. And in fact, if you look in detail at the microstructures that are found in these things, the nice layering is this part when it was underwater. And when it's stranded and covered in air, you get a more degraded biological community forming on them. And you don't get the good layering anymore. So it's turned it on its head. The club-like stromatolites are not only not diagnostic of intertidal environments, you can't even form them in intertidal environments. The typical stromatolites that form in intertidal environments are actually flat laminae without any relief. And that's shown by reconstructions. There's a reconstruction from the Proterozoic from Canada of a particular formation, showing you the different morphologies and how they vary with water depth. And in very shallow water depth, and you don't have this synoptic relief anymore. And so I think this applies to the stuff that I was doing in Scotland. So here, for example, we've got a domal structure, and then the domals, that dome dies out upwards, I think, because the water level is very low then. And then I found more identical fascies in East Greenland, and another example here where there's a domal structure that's forming on top of an clast, an eroded clast that's been disturbed by a storm that's sitting in there, and the stromatolites grow in it and form a dome, but then the relief gradually diminishes upwards because they're in an intertidal environment, and you can't form that relief there easily. However, having shown that other people are wrong, I must confess I was also wrong, because in the Scottish rocks I saw these round things here, which Roy McGregor from St Andrews said these are anhydrite nodules, just like we find in the Persian Gulf. But because I was so sure that it wasn't like the Persian Gulf, I said no, they're not. They're metamorphic nodules. But then in Greenland, where you get the same fascies, you have anhydrite relics inside things that look just like this. So clearly I was wrong about that. But these anhydrite nodules in this case were forming in subtidal conditions, but that association was there. It's an arid shoreline. These are some more of the Greenland ones. They've got beautiful little microstructures here. And one of the things I was able to do with these, because they've not metamorphosed, one thing that happens during metamorphism is that potassium feldspar reacts to form metamorphic minerals. And so you often lose a lot of the potassium feldspar. The potassium feldspar luminesces blue if you shine a beam of electrons on it. And so the little blue things we've got here are little grains of potassium feldspar. So it's showing us where the detritus is, where the sand and silt grains are. And so there's the layering that we see in ordinary light. There's the view in luminescence that shows us where the detritus is. And it's picking out layers that are richer in detritus. So it's illustrating the extent to which there are actually detrital layers in these stromatolites versus just precipitates of carbonate. Now then, Andy Knoll. I'm introducing Andy Knoll here because I started working with him because he was the successor of Barkhorn in Harvard, and he was finding microbial fossils in Precambrian rocks. So I went to visit Andy in his office in Harvard, a great big office, as you might expect, and in the corner was a safe. Obvious question, why have you got a safe there? He said, oh, well, Barkhorn had it for the moon rocks. Wow, this was about 1990 that I was there, and there was some time since the moon rocks had been there. So that was very, very impressive. Well, why is the safe still there, Andy? It's waiting for Mars. Well, he's still waiting, but it may be that in the next 10 years or so, there might be some rocks retrieved from Mars. Anyway, he was thinking in the longer term. Well, he's just retired now, so I don't know if he'll muscle his way into that. Well, he is a great expert on Precambrian paleobiology. There's Barkhorn, and there's the moon rocks. And these are some of the marvelous microfossils that Andy Knoll worked on. I mentioned earlier on, I had a picture of Svalbard, and there was a thing called the Draken Formation, and it's got these exquisitely preserved microfossils in silica. So this early diagenetic silicification. These are the coccoid cells, they're the filaments. This is a field view. It's showing you a breccia, like an intertidal breccia, with some black silicified fragments and some white that are not silicified. And when the silica comes in, it comes in early, and you often get these beautiful preservation of these structures. So it's this sort of environment. It's like a supratidal environment with springs discharging, and then a shallow lagoon next to it. And one of the most bizarre things was this structure. So one of my samples had this in it. I never in a million years would I have guessed what this is. And it turns out that this is a fossil that had just been discovered in the modern and the Bahamas. But the fossil one was described first before the modern one. And it's a cyanobacterium, but it's quite a big one, and it's forming like a tubular structure. And here it's actually been calcified. So dolomite's precipitating. It's very unusual to find that in the Precambrian, very unusual. Still not entirely clear why, but where it does occur, you're actually seeing the body fossils themselves. And this one, this one is diagnostic actually of an intertidal environment. So there you can see some of the calcified cells preserved in dolomite. Here I've shone UV light on them, so some of them are fluorescing brightly. So you're getting these tangled mats of filaments, and it's separated by the zones that have got cement in them. So just wonderful, wonderful preservation. And in the nearby subtidal environments, you get things that are not really quite stromatolites, but but nevertheless, it is trapped sediment. So this looks like an ordinary sediment, but it's got these vertical little filaments in it, which may be algae rather than bacteria, just trapping the sediment. So it's a bit like seagrass trapping sediment in that sense. And then these are some of the microfossils that are getting squashed in there. So just quite extraordinary preservation, but it's very rare to see it as good as that. Now a little bit about Conophyton. So I've talked about how the morphologies are very different and difficult to understand. Well, the simplest one of all is Conophyton. And this is a simple conical shape. And these things occur, there are dozens and dozens of them that can be seen in outcrops. And they would have formed like a nest of rocket cones on the seafloor when they form. Something really very, very distinctive. So how do they form? Well, there's been some progress here. And one thing that people have done is to do some theoretical models. And this is an example of a paper published in a physics journal about stromatolites. And they said, well, you know, if you name a parameter, which is the rate at which the microbes want to go upwards towards the light, that's the phototactic response. And you say, well, how fast is that happening versus accumulation of material perpendicular to the layer? So that could be, that accumulation could be sediment trapping, or it could be mineral precipitation, but something going on that isn't biological, not directly biological. So how fast is the one happening versus the other? And they say, well, if you get the sediment accretion dominant, then you'd get a domal shape. But if it's the phototactic response that dominates, what you'd expect is the cone. But there's one more thing that you need. You need a way of stabilizing the shape. Otherwise, it will just collapse. And there are some modern lakes where there's some very small possible analogues of these things. See, that's only a couple of millimeters across. There they are in 3D. And these little structures have clay, magnesium silicate clay, precipitating in the EPS, so in the microbial mat, and that stiffens it and allows them to keep their shape. Now, we know that actually magnesium silicates form quite readily in Precambrian rocks. People have been, in recent years, they've been realizing this and finding examples of them. So and in fact, people have often remarked that the conophytons do have a bit of clay in them, whereas in between the cones, you don't have clay. So that always was a bit suspicious. So now what people are saying now is, well, this clay is not a sediment, it's actually precipitating from the seawater inside the EPS. So you see there's been some progress there with conophyton. They are the archetype of stromatolite in quiet water, deeper environments, where it's just basically the pure growth with a bit of mineralization of the mat that's occurring without all the complications of sediment accumulation. Right, I'm going to take you to Mauritania to see some more examples of these sorts of things. So this had a chance to visit there in the late 1980s and to work on material that marvelous French woman Janine Sarfati had been working on. So in the fringes of the Sahara Desert there, you get these amazing stromatolitic biohome layers. Well, there are biohomes and biostromes forming a whole series of layers within this Neoproterozoic succession. And here are some of the conophyton. There's quite a big hammer for scale. So these are really quite enormous things. But in addition to having what you might call the normal conophytons, there are also these are called jacutophyton, and these are really weird. So there you've got the rocket cone thing, the conophyton thing, but then what's this? What are these things sprouting off the side? So if you reconstruct that in 3D, you've got like a rocket cone in the middle, and you've got a series of petals around the outside, which are growing off sideways. Now, you know, I worked on these things. We published a paper about the diagenesis and so on, but I have not got the foggiest idea about how such a thing could possibly form. A few years later, Linda Kay and colleagues worked on this, and they actually came up with a model about how actually alternating quiet water environments with more turbulent environments where sediment formed, and then quiet water environment came back. That could actually lead to a situation where you start when you have sediment stromatolite growth restarting, it starts at several points on the cone, and you then get these individual petals growing out. So they came up with an environmental type interpretation to explain how they could form. So something that just seemed impossible, couldn't think of any explanation. At least they've come up with an explanation that may have some merit. And people have continued to think about these theoretical models, what you can do. And in this case, they had a theoretical model involving sediment input and how that will be distributed. So that can be useful, but it still doesn't get anywhere near explaining all the different distinctive shapes that the Russians identified back in the 1960s. Now, it's actually quite useful to do this sort of theoretical analysis when it comes to thinking about, are things that are very old actually biogenic, or are they inorganic? So if we go back to the Archean, 3.4 billion years ago, people would say, well, was there really life back then? So we get these things, we get these structures, but are they a sign of microbial life? Or could you just get them forming inorganically? You get crystals forming, get sediment coming in. Why does there have to be a biological influence? So this is something that people have thought about quite a bit, and they've used some of these theoretical models, and they've looked quite closely at the complexities of shape that you get in some of these structures. And the conclusion is that they are indeed biogenic, that there are things that cannot be explained by purely inorganic processes. So, and that's now been corroborated by more recent studies that have also found microbial cells within some of these structures of this very ancient age. And I think this is the last example I'm going to show you, which is one of the weirdest of all, and that is the manganese nodules. Now, you may have seen in the news that there's some disquiet about the idea of hoovering up manganese nodules on the deep sea floor to get at the cobalt and nickel that they've got to power our, electric cars or whatever, because it would be an environmental disaster. And one reason it's an environmental disaster is because these things do not grow very quickly, and yet they look like stromatolites.
Is that in fresh water or seawater?
There's a deep sea. Yeah, so in the deep ocean, deep Pacific Ocean, several kilometers depth, there they are. Now, how fast do they grow? Can you give me a guess as to how fast they grow?
Several years.
Several years, there we are. Right, so in 1 million years, they would grow, and we know this from geochemical studies, in 1 million years, they grow 1 millimeter. So if you mine them, they're definitely a non-renewable resource. But can something that grows that slowly be organic? Can it be microbial? Or must it be inorganic? The jury is out on that one. So a promising way forward is to do more and more experimental work. And I showed that picture earlier on, and these guys are doing some very interesting work growing mats in the laboratory and seeing what they do. But nevertheless, these problems of macromorphology are very, very difficult, very elusive, difficult to solve. I just listened to a nice podcast yesterday by Martin van Kranendonk. It's in a website. There's a website called Geology Bites. Have you heard of this? geologybites.com. And this is the latest one that's been put up on that website. And he's got it's a really nice, sort of up-to-date account of a researcher using all sorts of techniques to find out about some of these really old stromatolites. And their interest there, because he works on Martian imagery as well, and he'll obviously be wanting to look at rocks from Mars. So they're using that as a comparison, the Archean Earth versus Martian Earth, to look at that. So that's a very interesting podcast. I looked on Amazon, and what can you buy on Amazon? Well, you've got for, how much is this? $11,000, or no, sorry, I'm not sure what currency that is. It may not be such a frightening currency. It's thousands of something to buy that book. And then what about this one? Blame It on the Stromatolites. Real world creative thinking for young adults. So that looks quite a nice Christmas present for somebody. And that's the end. Thank you very much.
Right, yeah.